my name is Triona O'Connell and I'm a BioAT funded researcher with the School of Biotechnology in Dublin City University, where I work very closely with the Irish Separation Science Cluster. All of that's a mouthful, but my PhD is a little bit shorter. I'm looking at sugar-coated cells. So the ISSC developed probes, and what these probes do is they can detect sugars, and these sugars are usually found on the surface of proteins. So when I started working with them, I said, where do we find a lot of proteins? Where do we find a lot of proteins with a lot of sugars? And so we thought about this for a bit, and we said, do you know where you find them? You find them on cells. So I look at cells. So every single cell in your bodies right now is covered in sugar, and it has a number of purposes. Sometimes the sugar is used to stick the cell to another cell. Sometimes the sugar is used by the cell to attach it to a surface. But another key way that it's used is by the immune system. So these sugar structures on the surface of cells can signal whether a cell is happy, whether a cell is dying, whether a cell is dead. And in some very specific cancers, you can even detect the sugar change. So to drop the cancer word in there, for a talk like this. So how do I use these wonderful probes given to me by my colleagues in the ISSC to look at cells? Well, I use a laser. Everyone loves lasers. So we use a thing called a fluorophore, which is effectively a dye that we attach to the probes, which we then attach to cells that display the specific sugar. Then I use one of my favorite instruments in the world, which is a flow cytometer, which passes the cells one by one past the laser. If the cell is dead, it lights up. If the cell is not dead, it doesn't light up because the sugar that I chose to identify happens to be gluconac, for those of you who want to Google it later or talk to me after the break. So after counting thousands of cells a second and collecting a lovely big data set for myself, we were able to show that these nice new probes that the ISSC have developed allow me to identify clearly live cells and to identify clearly dead cells, which is potentially useful in the laboratory, because figuring out if the cells that you're working with are dead is a pretty useful tool. You can also just check if the sugar on the surface of your cells change for other experiments that you might be carrying out. That's your experiment, it's not my experiment. We can also make beautiful pictures that are less beautiful on this big screen, but trust me, it's a beautiful image. They wouldn't let me leave the lab with it. We can use a very specific technology that colleagues of mine have developed to look at where the sugars are located on the cells using a microscope, and then overlay those on the same cell looking at a different sugar group on it, and get this theoretically beautiful image. Visit my lab, I can show you. So why does this matter? Well, it matters to me because I want to get a PhD at the end of the day. Don't we all? But it's also very useful in the real world. So if you think about the way a lot of the new drugs that are coming in, they're produced by biopharmaceuticals. And these are produced by cells, many, many cells in a bioreactor. The equivalent of an entire human blended was the best description I heard. And when that batch turns, you lose hundreds of thousands of dollars worth of drug. So if you could identify early on by checking for the sugar change, you could, in theory, save a lot of money. Now, whether or not the pharmaceuticals pass that on to you is a different matter, but it's a nice use in the end of the day. So, thank you very much for listening.